And then I always say, when structures are known, go and have a look at it. I mean, why not? It's all about this anyhow. These amino acids, the fold, how they interact, the specificity comes from there, the interaction of proteins. So these are two SMAD proteins. I think it's SMAD1, MH1 domain, that's the DNA binding domain, and how they interact with DNA. So this is DNA. This beta sheet here, this and this is a beta sheet, as you can see. This beta sheet is in the major groove and recognizes GC-rich sequences. Brinker also recognizes beta-rich sequences. There's no structure of Brinker, of course, doesn't exist in vertebrates, and nobody really cares all that much about it. So this is a beta sheet in the major groove. In this crystal structure, there was a second molecule that I also interacted with this small piece of DNA. It was probably a dimer binding sites, because we know there are 2-3 binding sites. Okay, so here the beta sheet, here how it interacts. Now you go to, this is actually quite fascinating. Now you go to another complex that has been solved where a trimer is in it. And this is here. Oh, no, this is, sorry. I have to go back. You can hear me and see me? Gasha, all good? Okay. Here. This is a trimer. It is a trimer or a hexamer. Nobody would argue against that. It's two SMAD1 or MAD proteins and one Medea, co-SMAD, so it's 2 to 1, but it's a trimer or a hexamer. This is the only structure available for a trimer. So look at this. It's a donut. This is one DNA binding site here, beta sheet. This is the other one. This is the third one. This is the trimer. There are interactions between each two monomers here. Same interaction here, same interaction here. So it's a trimer, but it's a donut. A hexamer would be twice this. How it would form the hexamer, I don't know, because the trimer is closed here. Maybe it opposes it to the other side. And the DNA, since this is the DNA binding site, would go right through this donut here. And I don't know, so nobody ever solved the structure of this. It could actually be that this thing then really wraps around the DNA like a double donut, like a hamburger with a hole in it, I don't know. As I say, I think there is nothing known about this about DNA trimer hexamer structure. But this would argue that this dimer, once it's there, then it's sort of, the DNA would be in the middle. So the trimer would go along the DNA molecule and fall into where the DNA binding site is and then be locked in. Very interesting, very intriguing, but not really known, because people nowadays with cryo-EM, they would rather love to solve big structures, and small structures are not so popular anymore, and this needs DNA plus, and so it's a bit complicated. But answers, a lot of cool answers will come out of this. So although we sometimes think this field has been sort of, we are done, I think we're not done at all. The interesting details and how it really works and is so specific, that remains to be done. So we have 20 minutes, 25 minutes for a 1-hour lecture, but we'll do it because, as I also realized, this lecture is a bit longer because I go into a bit of methods like mitotic clones, but we don't discuss mitotic clones this year anyhow. So you'll get a sort of an intellectually not light version, the same version as last year, but experimentally a lighter version than before. And then we hope to keep in time, which is quite important for you and for me too. There's nothing like never knowing when you're going to be off the hook. So we try to keep in time and give over to Gustavo at 3 o'clock. And the break will be hopefully at least 10 minutes, probably 15. DBP signaling pathway. Can we still learn something? I mean, we've told you all of this with Schnurri, MAD, Medea, Brinker, and so on. Yes, there are a lot of open question about the ligand. How does the ligand disperse in tissues? How do cells respond to different types of doses? DPP is a morphogen, and morphogens are very important because the same molecule at high doses turns on one gene class, lower another gene, and yet lower another gene. Same pathway, same components, but depending on the level, you get 3 different response genes. So this is clever, because to turn on a gene, you need something. But if you can turn on 3 genes in different places with exactly the same thing, well, good enough. Maybe as you make a DPP gradient, you have more and more brinker the further out you go. In the middle, brinker is completely repressed, and then you make a bit more brinker, a bit more, the further away from the source. And brinker is a DNA binding protein, can also be used to integrate DPP signaling. So here's the pathway. Brinker, we have a cell in the Drosophila embryo that doesn't see a DPP signal. It has high levels of brinker. Brinker binds to all DPP targets, make sure that they're off. Now, this cell, maybe if it doesn't see DPP signaling, it will stay like this for hours. Some cells in Drosophila in the embryo, they stay like this for the whole of embryogenesis. How do you find them? Well, make a brinker GFP transgene, put a GFP instead of in the brinker locus here. And then you see, okay, every cell that is green has no DPP signaling. And if a cell, you can make a movie and then you can count how many cells have never been not green. Then you know, you have to take an unstable GFP, but all doable. So this cell stays like this forever. And now, maybe it's a midgut cell, it's close to the DPP secreting cells in the midgut. The midgut comes together, DPP is expressed, and now the endothelial cells, the endodermal cells, they see it. Poof. In order to turn DPP-dependent target genes on, you first need to remove brinker. How do you do that? You recruit on the 15 base pair elements, which has 3 SMAD binding sites, probably 2 for MAD, 1 for Medea. These 15 base pairs recruit Schnurri once MAD Medea are bound. Schnurri acts as a repressor. Brinker protein is repressed and then slowly degrades away. The protein is still there, so you have to degrade it. It's a very unstable protein. So as soon as MAD Medea come into the nucleus, they're associated with Schnurri on these elements, brinker goes away. And now DPP target genes can be activated. As it turns out, and this is true for hedgehog signaling, winglet signaling, and so on, they all have repressors here. CI is the repressor of the wingless pathway, CI activator, non-processed form, then activates. They all have this switch. They all have a protein in the nucleus, which is here Schnurri, which helps to repress the pathway, and then is a switch from repression to activation. Often, these DPP target genes here, they don't even have to be activated and have MAD Medea binding site. The not being repressed by brinker here is enough to activate them. So perversely enough, the most important thing the DPP pathway has to do, question, chat. No, anyhow, I don't see the question. The most important thing the DPP pathway has to do is together with Schnurri to repress brinker. And this is quite perverse. So we made experiments where we removed, if you remove brinker, well, it's not such a strong phenotype. But if you express brinker in the whole embryo, even in cells that have seen DPP signaling, it's a no-go, no DPP targeted anymore. Schnurri, no gene can be activated on anymore. So this branch of the pathway is by far the key branch of the pathway. It's perverse, but this is how it is. Completely unexpected. We did not ever think about this. And even more surprising, in vertebrates, nothing is known about this pathway here. And maybe this is particular for flies. I don't think so. And maybe in flies, in vertebrates, there are several Schnurri genes. There are 3. Maybe there are other factors that take this thing here. You cannot define enhancers the same easy way as you can do in flies. So this is still completely open. Some people even argue that BMPs cannot repress transcription. directly. I mean, mad media can directly repress transcription. That's what they do here. It's only when they bind with Schnurri they repress transcription. So it's a direct repression. Without binding, no repression. Okay, so this is the pathway. If we think about morphogens that have effects at high doses, medium doses, and so on, then this system here is useless, because here we don't only have no and high. And the morphogen distinguishes between these two situations. That's the interesting of a morphogen. A morphogen is not interesting if it's there in full dose and there in no dose. The interesting thing is a morphogen is that as you go away from the source, you get less and less. And these are the important things, not the on and off state. So what we actually want to learn in most of these signaling pathways as they work as morphogens is what happens between these two states. These are the off, but every cell is off if it doesn't see a morphogen. And this is the high level that has nothing to do with morphogen, that's the adjacent cell. So what happens in between? What are the important things that happen in between when molecules are read out as morphogens? So the tissue, we look at it here is the wing, and I'll give you a wrap-up of this, and then Gustavo will take some of this information with you. This is kind of a bit complicated, but he will go back and redraw some of the important things. This is a wing, a Drosophila wing. Unfortunately, you haven't seen many of them in real life. It's a beautiful structure, has some veins. Here is vein 1. Here's vein 2, here's vein 3, here's vein 4, vein 5. There is a compartment boundary here at this place here, close to this vein here. So whatever cells divide, they never populate. If they are born in this part of the wing, they're part of this, they will never grow into the other area. So it's a compartment boundary means cells don't mix. They make a very straight boundary. It's like oil and stuff. They don't mix. And therefore, whatever happens in one compartment does not matter necessarily to the other compartment. A cell clone, yeah, Gustavo will go into this. Why was DPP so fascinating? This is the wing phenotype when you remove DPP. This is not an S allele, this is a D allele. There are many alleles of DPP. S4 lacks expression in the gut. D8 lacks expression in the wing imaginal disc, has gut expression. And D12 also lacks expression in the wing and has gut expression. As a trans heterozygote, they give rise to these wings. So DPP is required to make the wing for growth and probably for patterning of these veins. If you ectopically induce DPP, you make a US DPP transgene, and you activate GAL4 in a few cells, let's say in this area here, you get a wing duplication. This wing duplication only duplicates part of the compartment you have induced DPP in. So you cannot, by expressing DPP here, you will never affect the other side, but you affect the duplication in posterior, this is posterior, and anterior. So here you see you duplicate, you have wing 5, vein 4, vein 5, and then you go back into the wild type 4, 5. So this is a duplication. You kind of know this from the book, from the earlier developmental biology session. All these slide shows DPP is required for the wing, and if you have too much DPP in two sources, then you duplicate the entire structure. So it induces growth of an entire duplication, and it is required for growth of the wild type structure. And then the question is sort of, where is DPP expressed? But DPP is expressed here in the center. That we know now, if you go to the wing imaginal disc, it's expressed here in a stripe. So now for this lecture, we don't deal with expression in the visceral mesoderm, which induces labial. We see that DPP is expressed in the middle of the wing imaginal disc. This structure will give rise to this. It's a complex 3-4 dimensional refolding that maybe Gustavo will show. It's not so important. We only deal with this here. This is anterior, this is posterior, compartment boundary. That's how DPP is expressed. How can it influence the development of the entire wing on this side, which is over here, and on this side, which is over here. So two simple questions we want to address. How do you make a stripe of expression here that is very stable? This structure is very little, about 30 cells, and grows to 50,000 cells during larval development. DBP is always a nice stripe in the middle. How do you do that from a very small to a very big structure to always keep this stripe nicely in the middle? And how do cells then respond to the DBP ligand? DBP ligand is only made here in the center. So it presumably makes a gradient on this side, a gradient on this side, and then the gradient instructs where you make vein 1 and vein 2, and on the other side where you make vein 5. So how does that work? Here we don't deal with DBP off and DBP on, but DBP graded and different responses. That's what we want to see here. Morphogen, there are many morphogens. Every tissue is patterned by a morphogen, and in no case we know how it really works. Is there a gradient? How sharp is the gradient? How far does it go? How do cells respond? We know a bit here and there for each of these morphogens, but there's no conclusive view. So how do you make this stripe of expression of DBP? How do you make a stripe that remains in the middle of a structure that constantly grows and becomes maybe 50 times bigger during its growth? This is kind of a cool thing because you grow, but you want to have a stable compartment, anterior, posterior, stable line of DBP in the center. If it would grow to the other side, it wouldn't work anymore. And what people found is that there is a transcription factor called engrailed. It's a homodomain protein. It's expressed only in the posterior compartment here. Always from the very beginning, it's probably autoregulated. Cells don't change, fade. These cells are engrailed positive. These cells are engrailed negative. The boundary here is a compartment boundary. Engrailed positive cells will never grow into this compartment, and engrailed negative cells will never invade this territory here. There is an absolute strict border. This border in the wing lies somewhere between two veins. It was very puzzling. A good friend of mine, Ginés Morata, discovered this compartment boundary first when he made clones with forked. Martin told you yesterday. If you make clones with forked, you see a clone here, but clones at the compartment boundary, they never go from one compartment to the other. And then he postulated that these cells are unmixable, and they're cells that are born here, they will always be here. They make all kinds of clones, but never come to the other side of this imaginary line here. And this is a concept, this has been proven right. And it has been shown that if engrailed is expressed in these cells here, then they will never go to the other side. And there is a physical boundary, there's actomyosin there that sort of pulls these things. So the cells have different properties and they can simply not invade the other cells with the other properties. So it's a compartment boundary, no mixing, like water and oil. Now we were asking how you make a stripe here. So how would you make a stripe here in this area? If you already know that you have from the very early embryo on, you have cells that make engrailed and cells that don't make engrailed. So how you could make a stripe is to, you can think about it. I don't know how many ways they are. You could say DBP should be repressed by engrailed, so it cannot be anywhere where engrailed is, and then you don't have a stripe, but you have a region here. And then you could say maybe if engrailed would secrete a ligand that itself goes into the other side and makes a gradient, then you could actually make a stripe here. Repression here, activation by a secreted ligand from that side. And then the first stripe, the compartment boundary, would always maintain this other stripe because proteins come over, activated. It's a beautiful, simple system. And of course, I wouldn't tell you this solution if it wouldn't be the solution. So should I really go into all of this with you? Yes, I do. You see, this is the problem now a bit with not having a blackboard, because here I used extensively the blackboard to show you. So here is the question. This is the stripe of DBP. I tell you now that all of these cells here, over here, they make engrailed. That's what I just showed you. So then the question of how to make this stripe becomes easy. We can say engrailed doesn't allow DBP transcription, and then DBP can only express to the other side. And as I said, if you have a ligand from here that disperses, but it of course doesn't disperse to the other side, this is a short-range ligand, then the stripe size would be maintained all the time, because you have a ligand made here, it will disperse. And the structure grows, but the dispersal will not be increased as the structure grows. Dispersal is dispersal. And that's actually what happens. So, I have to go to mine. Next year, this will be a fantastic lecture. Today, I will have to improve a bit. Let me go to the share screen whiteboard. Oops. Eraser. So I'll explain it to you here. Gustavo might use a bit more things. I think it's a nice concept because this concept is used again and again and again, and it shows you also that some pathways can be different. Compartment boundary, cells, this is posterior compartment, this is anterior, and the DPP stripe is here. These cells, okay, these cells make DPP. Go here again, and this is the compartment boundary, exactly at this line here. Ooh. No posterior cells make DBP, only a stripe of anterior cells. These cells make engrailed. And engrailed can repress transcription. It's a transcriptional repressor. But it doesn't really regulate DBP. What engrailed does, it represses CI, cubitus interruptus. If you remember from the block course, cubitus interruptus is the mediator of the hedgehog pathway. It's a transcription factor required for hedgehog gene activation and also for repression. Cubitus interruptus. Okay, I mean, this is DBP signaling here. What about hedgehog signaling? But what engrailed also does, it activates hedgehog, the ligand of the hedgehog pathway, in each and every cell. So now you have, this is the mediator of the hedgehog pathway. This is the ligand of the hedgehog pathway. These cells in the engrailed domain cannot respond to hedgehog because they lack CI. CI is the transcription factor of the hedgehog pathway mediator. But they make the ligand. So here you have a cell population that makes ligand, cannot respond to it. And the other population makes no ligand, but can respond to it because CI is there. CI is not repressed because CI is repressed by hedgehog. So here we have a situation where a group of cell makes a ligand, but blocks the pathway of the ligand by negatively regulating its transcription factors. So the ligand will disperse. The ligand will go out of this domain here because it's made here. It will go over there. It can activate the hedgehog pathway. So CI activates DPP under high levels of hedgehog. Hedgehog disperses. Hedgehog signaling leads to not proteolytically cleave CI anymore. You have CI in the nucleus here, and CI will activate DPP. This structure can now grow or not grow, less or more. The stripe is always there. The cells don't mix because they have engrailed. And the stripe, you have two non-mixing populations. You can make them as big as you want. And there will always be a nice stripe of DPP. And that's where DPP gene is just transcribed. That's why it's transcribed here. It is not transcribed here because these cells don't respond to hedgehog. It's not transcribed out here because hedgehog doesn't go that far. And then you make DPP transcription here, and then you make the DPP protein here, and then this DPP protein makes a gradient, which is high here and low here, and that's how you pattern the wing. And this has to do with this two-cell population. This boundary here is already made in the very, very early embryo. The wing imaginal disc primordia, which has about 10 cells, I would say, maybe even less, is born around the compartment boundary and already brings along engreled expressing cells and engreled non-expressing cells. Maybe 4 expressing and 6 non-expressing. This is a bit smaller than this. And from these 10 cells, it grows, it grows, it grows, it grows, and DPP stripe is then turned on in the middle. Even if it grows, the DPP stripe stays there. That's the textbook model, and that's what you need. That's what I can tell you. Shinya will tell you a bit more. So let's briefly go back to the... So what about DPP then? We said here in this model, engreled turns on hedgehog in the engreled cells here. Engreled represses CI. So these cells don't respond to hedgehog because they lack CI. It's like a cell cannot respond to DPP signaling if it lacks MAD or MEDEA. There is no cell that has no MAD MEDEA. The DPP pathway doesn't function in this way. DPP's pathway is a nice pathway. It allows every cell to have a receptor, make MAD, so all cells can respond. And in hedgehog pathway, many cells cannot respond to the pathway by itself. Is this logic or not? Is this a different logic? We don't know. But pathways have sort of inbuilt logics that we don't really understand. Maybe hedgehog is membrane bound, and membrane bound ligands, they actually want to be exposed to the neighboring cell and not to itself. So you don't want to respond it if you have a membrane bound ligand. I don't know. This is actually possible. These 2 slides were supposed to show you the hedgehog pathway, but all to say this is certainly not a question at the exam. So just listen, don't try to remember everything. So briefly about DPP, because we want to finish with this. The morphogen function of DPP, oops, I think I left you now. Share screen here. 5 more minutes. I go relatively rapidly through this because, so this is the concept of a morphogen, high concentrations from the source. So this is the anterior stripe of DPP. In the posterior, there will be less and less DPP, and some genes are activated in different doses. And the question is, how does that work? Why do these cells respond to high? Why do these cells respond to medium? How do they do it? And so on. These are the questions that you have to ask beyond signaling for a signaling molecule that is a morphogen. In order to investigate that, we of course needed genes that have these expression patterns, high, medium, low. And very early on, there were 2 genes that were identified by markers, enhancer trapping. One was called SPALT, another was called OMB. And you can see SPALT is not so broadly expressed. So this is a gene that is only expressed at high levels of DPP, if it's regulated by DPP. And OMB is much broader expressed than SPALT. So OMB would be down here somewhere, responding to lower levels. It is known that OMB and SPALT determine these vein positions, vein 2 and vein 5. So they were very good candidates for the genes that respond differently to different levels of DPP. Now, what do we know about these different responses of OMB and SPALT to different levels of DPP? That's the key question here. Why is OMB more sensitive? Does it have more MAD binding sites, or are they differently spaced? Or that would be the molecular explanation. We need a molecular explanation for why they're differently expressed. Both need DPP. So I can give you kind of a, since I said to test this, you need loss of function. Are they dependent on DPP signaling? Do they need the receptors in all the cells to respond? And that comes with clonal analysis that we do not do. We classically did Markov analysis, but we skipped this. We cannot show you much clonal analysis. Gustavo might go a little bit into this, but I'll skip some of these. These are the experiments. I skip the experiments and I give you the take-home message. So you see, this is Markham clones and mitotic clones. That's also another question at the exam, pretty sure. Anyhow, so the take-home message is here. I should go, no, I go back here and then I jump again, sorry. So there are two. It was pretty clear for us when we started that Spalt has MAD Medea binding sites, maybe many of them, or maybe not many of them. Let's put it this way. OMB has probably many, so even if you have little DBP, little SMAD, there are enough SMAD binding sites, so that was the one. OMB has many SMAD binding sites of high affinity, so it can read out even very low levels of DBP. Spalt has few sites, so it requires high level of DBP. That's why it's only expressed there. But now think about Brinker. Didn't we just say that actually the whole system works very differently? It's actually Brinker that counts. And for many, I mean, for 10 years or so, we thought it's the MAD SMAD levels that is interpreted on OMB. We tried to map SMAD binding sites on OMB, but we didn't find any. And the ones we found, we mutated them, there was no effect. So after many years, we realized it cannot be the SMAD input that regulates the expression of OMB and makes it different from the expression of Spalt. But what is the other logic candidate? Brinker. Brinker expression is low in the center, and then slowly as DBP goes up, Brinker goes up here. As DBP goes down, Brinker goes up. Here you have enough SMADs to repress Brinker. Here we have no SMADs, Brinker is on, and of course in between it's graded, but it's an anti-gradient. So the easy solution would be is that OMB has Brinker binding sites, and these Brinker binding sites are of low affinity or of high affinity. You only repress OMB by Brinker at high dose. Spalt has Brinker binding sites, and if you change the affinity of these sites, here these are high affinity sites, so you don't need much Brinker to repress it. So the whole system went from a MAD-centric to a Brinker-centric. And we did all these experiments. We mapped the Brinker binding sites on OMB, we mapped the Brinker binding sites on Spalt, we mutated them. If you map the Brinker binding sites on OMB, OMB is everywhere. It's not repressed anymore. So the input of the morphogen doesn't come from MAD Medea, but it comes from Brinker. The Brinker input is, of course, exclusively controlled by MAD Medea. If there's no MAD Medea, Brinker is high. If there's lots of MAD Medea, Brinker is gone. And in between, you find all the other levels. So you first make a Brinker antigradient, and the Brinker antigradient regulates OMB and Spalt. And these we could do with biochemistry and mutating sites. So if we go back to the very last slides, here we have the logic again of the pathway. When it's off, Brinker is everywhere. Brinker binds to GC-rich sites. These are the SMAD binding sites. And represses transcription. In the on state, there is no Brinker, and MAD Medea can bind to these sites here. And then you activate these genes. But what MAD and Medea at high levels mostly do is they combine with Schnurri, bind to these are the 15 base pair elements, they're called silencer elements. That's where MAD, Medea, and Schnurri bind, and take Brinker away. This is the important thing. It's the regulation of the levels of Brinker that determines the expression of the target genes. And that's very surprising because the morphogen is read out at the single locus. That's Brinker. And this locus then translates the graded readout into the rest of the cell. So you have a morphogen sort of a morphogen sensor. These brinker binding site, these mad medial Schnurri binding sites here in the brinker locus, it's like if you push them into a cell, like the oil in the car, the oil measuring device in the car, you put them in, you take out, and you ask how many mad medial Schnurri are bound to this, and then you know, oh, I'm here and here in the gradient. So this last slide, this is how the gradient works. The graded readout of the DPP gradient here is first translated into an inverse gradient of Brinker, and the cell measures the inverse gradient of Brinker. The target genes like OMB, SPALT, they don't have MAD binding sites, they have Brinker binding sites. Why there is this indirect translation? Does any BMP gradient in any vertebrate organ work directly or also indirectly like here? We don't know. But in the fly, it's pretty clear. The gradient generates an antigradient. The antigradient is a transcription factor antigradient. And this one either binds to target genes and represses them, or binds only to one target gene, represses it and not the other. Oops. Or doesn't, when it's gone, it doesn't bind anywhere, then these things are on. So they're not activated, OMB is not activated by the pathway. It's de-repressed by the pathway by removing the repressor. OMB is repressed, and here OMB is not repressed anymore. So the logic, molecular logic, is very different to what we would have thought. The nice thing is that the morphogen is read out at one single genomic locus, which is the Brinker locus. And Brinker has 10 such binding sites for mad media Schnurri trimers. Connie Basler tried to remove some of them because we thought evolutionary, you make bigger wings, if smaller wings, depending on how many sites you have. That was early days, no CRISPR. So we should go back to some of these experiments and do them with CRISPR, nicely defined. But that's at the moment where we are. Read out the gradient at one locus, Brinker locus, and the Brinker protein is then the one that is measured in the cell at these different enhancer levels here. 15-minute break. And then Gustavo will take over and hopefully not keep you busy for too long. If you have questions,